Okay, so we're recording, and Byron, I'll go ahead and introduce you. Uh, Byron was recommended to us from Ellen Crocker, Dr. Crocker at UK, and Byron's located, I think, around Virgie in Pike County, and he's got a pretty broad background in mushrooms, which is the subject for tonight. And Byron, I believe you work at UPike, is that correct?
Yeah, that's correct. I'm an assistant professor here.
Okay, good deal. So share with us any other introductory information you want to share, and then you can get started.
Sure. Yeah. So, I think you covered it pretty good. I'm originally from Martin County, went to Pikeville College, then Pikeville College for undergrad, changed to University of Pikeville while I was there. Um, and I told them my senior year, I said, if you all will just pay me instead of me paying you, I'll hang around. It took them a year or two, but they took me up on it. My background was originally in botany and ethnobotany. So I've done quite a bit of work in that. And then from ethnobotany, I made the jump to fungi through lichens. I call lichens like the gateway fungus. It's what tempts botanists over to mycology. So I went through that route and studied a lot of bryophytes, pteridophytes, and lichens in Pike County. Um, and then from there, done my graduate work in entomopathogenic fungi, so the zombie forming fungi. Um, and now, uh, most of what I do at UPike revolves around teaching those organismal and environmental courses, and running a couple of research labs, which we'll get into. And I have a PowerPoint here. Uh, I'll send the request to share my PowerPoint. All right, can you guys see this?
We sure can.
Okay, great. We good to go?
Go for it.
All right. So, mushrooms in the mountains. This is not an edible species here on the right of your screen. That's a bioluminescent species. This is a photograph I took 2 years ago, and that's Panella stypticus, the luminous oyster mushroom. There's some little coprinus here on the left-hand side, but when I start talking about fungi in Appalachia, a lot of people don't realize how many bioluminescent species we have here. And usually, when you say mushroom, people think of kind of earth tone, drab looking fungi. That's not necessarily the case. So, hopefully, by the end of this presentation, you guys will kind of appreciate the diversity here. So, we actually have Panella stypticus going through research trials right now. It has some active compounds, some alkaloids that it produces that may have some medical benefit. So, I'm working with a biochemist here at UPike to isolate those compounds. Right. So, what's going on at UPike, University of Pikeville, with mushrooms? A lot, right? My lab right now, I oversee 17 undergraduate researchers, all paid. I have a colleague that works with me that helps with this, Peyton Prater, and then we're actually in process of hiring another full-time lab manager position. And so we'll be close to 20 people in total working on these projects. We have a couple of big ones. We have a USDA specialty crop block grant to grow mushrooms in coal mines. I'm going to go into each of these in a little bit more detail as we move along. Mushrooms in coal mines, that's the first grant that we got, and we're about a year and a half, 2 years in on that particular grant. Last August, we received a National Science Foundation grant. It was called the eRise 2 grant, and that's in conjunction with Kentucky State, um, some industry partners, and a couple of other institutions throughout the state. This one is kind of a mashup between hydroponics and mycology. We'll get into that. We're currently in the process of putting in a 3-acre truffle orchard with a native truffle species, Tuber leonii. Oh, sorry, that was the old name. Tuber cumberlandensis. Sorry, the Cumberland Mountain truffle. It's a white culinary truffle, and we'll talk about that. And then we have a lot of other side projects going on. I just listed two. I don't think that I particularly talk about morel cultivation or wavelength cultivation trials in this presentation, but feel free to ask me. But essentially, we're working with Penn State to grow morel mushrooms outdoors, do outdoor cropping trials. And then this summer, we'll work on indoor cropping trials for Morchella mushrooms. And then the wave leek cultivation trials. This one is really wide open. When you think about plants, everybody knows at this point, if you're a gardener, or if you're, any kind of farmer, you're probably aware that different wavelengths, different colors of light impact plants' growth differently. But what we're finding out now is the same holds true for mushrooms. And so we're just in the discovery phase of that, which is pretty exciting. All right. So, what exactly are mushrooms? I get this question a lot. There's a lot of misconceptions around this, and forgive me, this may be common knowledge to everybody in this group. This is what I usually like to start with, so we're all on the same page. Now, this particular mushroom, this is another one. This comes from the Breaks Interstate Park. This is Sarcosypha occidentalis, the stalked scarlet elf cup. This is an ascomycota. If you actually look really closely, these little white dots, those are little bundles of spores getting ready to be released. A neat party trick for these, if you have students, is to pick one of these up and lightly blow in the cup. And if the fruit body is mature, it'll actually release a little plume of spores. And that always fascinates people. So, what are mushrooms? Ooh, it didn't actually do my. I'm supposed to have another thing here. Well, let me explain it this way. Mushrooms, there should have been a phylogenetic tree that just pulled up there, but it didn't pull up. Mushrooms are their own kingdom of life. They're not plants. And this is a relatively new thing. Up until 1969, mushrooms were still considered plants. So if you go to a lot of public libraries in the state and you look for books on mushrooms, if it's not a recently published book, it's very likely that you'll have to look for fungi in botanical books. But that couldn't be farther from the truth. Mushrooms are actually more closely related to animals than they are to plants. But they kind of got lumped in with plants early on because you know what? They're sessile, they're in the forest, they're not really doing much. And so we assumed that early on that they were plants. But they're actually quite a bit like animals. They breathe in oxygen like we do, and they breathe out CO2 like we do. And that's one of the things that we're always battling in our mushroom chambers, our mushroom farms at UPike is this delicate balance of what do we do with the CO2 that they're making? Because it's just like if you put a bunch of people in a room, the CO2 level is going to rise up and the mushrooms are going to, they may have ill effects because of high CO2. And as a matter of fact, if we look at this mushroom on the screen, this Sarcosypha, you notice it has this really long kind of stipe. Um, it's from CO2, basically, is what causes it to have these long stipes. In mushroom cultivation, we call this getting leggy. But essentially, what the mushroom is doing here is you can imagine, you're a fungal spore on the forest floor and you've germinated and you've grown enough that you're ready to make a fruit, a mushroom. Uh, and CO2 is denser than air, right? So it wants to fall down to the forest floor. You can actually see some pieces of leaf litter here at the base of this elf cup. So what the mushroom needs to do is it needs to get away from the high CO2, and so it'll actually stretch its fruit body to try to get it up and out of the CO2. Now we can utilize that for our benefit when we grow things like enoki mushrooms. When we want them to be long and leggy, we can do that. But something like an oyster mushroom, we wouldn't want them to be long and leggy. Okay. So what mushrooms are? Yeah, here's the slide. Sorry. And so you can see here, you have fungi and animals over here. So they're relatively closely related, on this phylogenetic tree, much more so than plants. Slime molds hold this weird intermediate ground, and I have a student that's almost ready to publish a book on slime molds of Pike County. They're another odd group that I find really fascinating. But this just to illustrate that fungi and animals are pretty closely related in terms of eukaryotic organisms. So, what do mushrooms need? Well, they need water, they need oxygen, and they need a relatively stable temperature. Now, those three things vary depending on what strain or species that you're growing. Some species like a really high humid environment, 95 to 98% humidity. They like it really, really, really high humidity all the time. Other mushrooms, like an oyster, do just fine at 80 to 85, and they like kind of these spikes in humidity where it gets really humid and then kind of dries out a bit for them. Oxygen is a big deal. like I said before, they're breathing this oxygen in and they're breathing out CO2. So they need a surplus of oxygen, but they also need air turnover. So you don't want the air to get too stale on them or that CO2 to build up. And so in a minute, you're going to see a picture of one of our environmental chambers, one of our grow tents. And I'll explain kind of all the pieces going on in there. But one of the things that we always try to do is how do you balance bringing fresh air in sufficient quantities, but not drying the air out too much, right? Because we need to humidify that same air. So we're always fighting these two. And temperature is a little bit easier. Um, you think of mushrooms as any other crop. Some crops you're going to plant in the late summer to go through fall. Some crops you want to plant when it's still cold outside. And so right now, um, we're actually planting crops that are designed to grow in a coal mine environment. And you may hear my 3-year-old in the background here. He's looking at me through the door. Um, but right now, our chambers run anywhere from 45 degrees Fahrenheit to about 55 degrees Fahrenheit. Right. So let's talk about the USDA grant. So with the USDA grant, there's a couple of things that we said we were going to do. Number one, we need to develop strains of fungi that grow in a coal mine environment. Now, what do mushrooms need, right? Well, they need stable temperature. Check. That's an easy one. They need a relatively stable humidity, and that's pretty high humidity. And then they need that air exchange, right? So really, the only thing we're battling in an underground environment is really the airflow and the humidity. And we'll see some graphs in just a second that show kind of what our data looks like for the coal mine that we're working with right now. Now, some other things that the USDA grant wants us to look at is so we're developing the strains. So it's a cultivar breeding program, and we've bred several cultivars that are unique to UPike right now, and several oyster strains. They're our own heirloom quality strains. But it also wants us to look at waste streams. So these mushrooms are a first phase decomposer. So, common misconception is that all mushrooms kind of grow on dung or feces. That's not true. Ours are a primary phase decomposer, which means that they're growing on cellulose and lignin. So, sawdust, soybean hulls, wood chips, logs, that sort of thing. Things like white button mushrooms would be a second phase decomposer, which they need things that have already been partially digested through once, i.e., the manure. So we're looking at waste streams. So far, we've successfully grown mushrooms on spent coffee grounds, spent brew grain from the distillery downtown, old textbooks and cardboard and shredded paper and straw. Basically, any organic compounds that we can add to it. Now, the caveat here is that we want whatever waste stream that we're using, we want it to get to at least 50% biological efficiency. And that just simply means, if I give it a pound of organic substrate, I get a half a pound at least of mushrooms back. Because right now, using standard industry accepted substrates, you can get anywhere from 80 to 100% biological efficiency relatively easy. So if I put in, 2 pounds of substrate, I'm getting close to 2 pounds of mushrooms back. So we want to try to hit that same benchmark. Now, growing underground is not a novel idea. Here is a sketch, and it's a sketch from the 1800s here. Let's see, 1857 from the catacombs of Paris. And so, people have had this idea before of growing underground. It's nothing that's too novel. Growing in coal mines is relatively novel, but people are growing underground. The number one cost, typically, is, besides labor, associated with a mushroom farm is your HVAC cost. If you're growing indoors, right? Because they're like animals. The more of them you have in a space, the more heat they produce. And you got to keep them the right temperature, right? The temperature needs to remain sort of stable. And so a lot of farms spend a lot of money trying to heat and cool. Um, and that's not really necessary if you have a space that does it for you. So here again, this is a picture of one of those, these out of business underground mines over in Paris. I call it a mushroom mine. I guess this was one of the catacombs here. But you can see these bales. Now they would have been growing white button mushrooms, right? And white button mushrooms are still the big dog. They're the leader in the industry in terms of volume of sale. Now, it's important to note that we don't grow any white button mushrooms. We're one of the only other mushroom cultivation labs in the country. And in terms of manpower, we're the second largest, only behind Penn State. And Penn State focuses almost purely on white button mushrooms. They do some specialty mushrooms, but the bulk of their industry um, research is in white button. So you can see here, it's got relatively good shelf height. Now, interestingly enough, they actually don't have to humidify this air at all in these catacombs. It's perfectly humid enough for the white button mushrooms to grow. So I don't have any pictures of our mine site. There's not a whole lot to look at, right? It's kind of dark under there. I mean, if you've ever been inside a coal mine, that's what it looks like, right? But I did just pull from the Energy Information Administration here, pulled a schematic of a typical coal mine. And this is basically what ours looks like as well. And so you have these long runways where they have mined the coal, and then you have the pillars in between. And so basically what we've done to start our research was we put sensors deep in the mines, various locations in the mines. These sensors are automated. They collect all the environmental data for us. That way we don't have to have students in the mines. We just have the sensors in there, and then once a week, we can go and get the readouts from those sensors. They'll just Bluetooth out to us. And so we can get that sensor data, and then we, in turn, plug that sensor data into our environmental chambers and match the conditions, right? So here is what one of the chambers looks like. This is the inside of the chamber, obviously. This is inside one of our lab spaces at UPike. And there's quite a bit of stuff going on here. Lots of DIY, as you can see here. What we've found is in order to replicate a lot of the environmental conditions exactly, we had to build a lot of the systems to do that. So when we look at this is a 10-foot by 10-foot tent. We have two of these. We have a 4-foot by 8-foot tent, a 3-foot by 3-foot tent, and then we have an 18 by 10 rigid walled walk-in structure as well in terms of our fruiting space. So when you look in here, you'll know, first thing I'll start with is this PVC pipe that runs along the ceiling here. What you can't see is this actually runs outside of the tent, and there's a large reservoir system sitting just outside the tent here. It's something like 170 gallons worth of water. Inside of that is a UV sterilizer, a float valve, and a pond fogger, like you might put in a koi pond. And essentially what happens is you'll see these black cords hanging here. These are sensors, and they're a bunch of them. They're all over the tent. There's a bunch you can't even see. But this goes to a humidistat, one of several humidistats. And what that does is I can program the humidity to match the coal mine. And so right now, that's what this was set to. The humidity inside of our coal mine was pretty high, as you'll see in another couple slides. And when the tent drops below that set point, the pond fogger just behind this wall will kick on. And then there's a fresh air fan that blows into the tote and it blows up into the pipe. So these pipes are always pumping in fresh air. That volume is something we have to dial in. And then when the humidistat kicks on, the pond fogger, you'll see that the air humidifies here. There's several nozzles, so we kind of get a blanket that goes over the entire growth chamber here. In the back, you'll see another. This is a 6-inch intake fan, again, bringing more fresh air in. What's not visible here is we have a couple of circulation fans, one over here, one that would be directly behind my right shoulder in this corner of the tent, because we don't want any dead spots. And this comes from just like knowing your space. You don't want any dead spots. You'll have spots where the air is not mixing quite as well, and you'll have dry spots and wet spots. And we want to try to make this as homogeneous as possible. Now, you see some mushrooms actually fruiting here. These are our own cultivars that we've bred for the coal mines. There's a couple species here that you'll see. We have some blue oysters down here. These don't have, this is our own genetic strain grown for the mine. We have some pinks that we're working on. Pink oysters are actually kind of a tropical mushroom. They like it to be quite a bit warmer, but we're breeding a strain that can handle the cold. Up here again, you see some more pinks, and you see some lion's mane up top here. Now, we had just done a harvest when this photo was taken. You can't see on the back here, we have some shiitake blocks in the back. This is a gold oyster block. Over here, we have some really big king oysters. So there's a variety in here right now. We grow somewhere in the neighborhood of about 17 different varieties that we're testing. And I don't know how many cultivar strains we're running at a given time in the lab. But I mean, probably 100 at least, different cultivars that we'll run the phenotypes out to test them in here. Here's some of the varieties that we grow. So we have blue oysters, we have some pearl oysters, we have gold, we have pinks. The oysters are really pretty. We generally always have these growing. These are the easiest ones to kind of get a pulse on what they want. So we always have these growing. We also grow lion's manes, other Hericium species. Here's a nice block of Piopino. Um, yeah, right. Again, some more Piopino. These are pictures from students. Students really like helping in the research lab. They all have their own little projects too. Um, I have an Anoki strain that actually come from Pike County. My Shiitake strain came from Virgie here. My lion's mane strain came from Virgie. Now, shiitake is not a native species, right? It's probably an escapee. We see that more and more, especially those gold oysters. And so we actually take extra precautions to not make sure none of our species are escaping the lab. Everything gets scrubbed. All the air gets scrubbed before it goes in or out of the room. So, all right. So some mine site data. What do the mushrooms need? Well, they need temperature, they need humidity, and they need oxygen, right? So let's look at these. Here's our temperature. This is in Celsius, but it stays pretty cool. And don't worry about the x-axis here. These are data points. This was early on. The data has remained relatively stable throughout. What we see is that mine is basically always between 40 and 53, 54 degrees Fahrenheit, which is absolutely perfect for our cold loving strains. Now, if I go back a slide here, you'll see these blue oysters here. This blue and this gold and this pink oyster, we can grow those in anywhere from 21 to 25 days from nothing, 21 to 25 days, and you're harvesting. But that's if it's kind of, 70 degrees, 65, 70 degrees. When it's colder, it takes quite a bit longer for them to get there. You're pushing maybe 30 days to get a harvest, but the quality is a lot better, right? You get a much better crop at the end of it. And 30 days is still a relatively quick cropping time. CO2 levels. Now, I should note that this is the CO2 levels in parts per million. Now, this is before they turn ventilation on. This particular mine was not an active mine, so there's no ventilation here. And so 1200 will work. It's getting to be a bit high for oyster mushrooms. I actually like to see it below 1,000 for oyster mushrooms. Now with ventilation on, it bobs around 400 to 600. I just don't have that data on here. Relative humidity. So mushrooms need relative humidity, not actual humidity. And so the great thing is the colder the air, the higher the relative humidity. And so we don't have to humidify the air at all in this space. Now with more ventilation, it brings these numbers down a little bit. But for our preliminary data here, we don't really have to change too much in terms of humidifying the air, which is really, really nice. And again, here, just another picture of the chamber. It's the same picture as before. Right, so here's the types that we grow. Now we also grow, multiple different varieties of this, just like you have different heirloom tomatoes, or I have different heirloom oyster mushrooms, right? Different blue oysters. So blues, pinks, golds, kings, pearls. I have black king pearls now, lion's mane, comb tooth, Piopino, chestnut, enoki, reishi, dried saddle, lots of cordyceps, maitake, shiitake. These are some biolumin. These are not edible strains. These are medicinal strains that are a little toxic. These are all bioluminescent as well. So this is the jack-o'-lantern mushroom that you see. Some people confuse that in the fall for chanterelles. Mycena chlorophyllus is actually a subtropical and tropical strain of bioluminescent Mycena. Same thing with this. This is the Australian version of our jack-o'-lantern. And then the tuber, this has now been renamed in the last month or so. It's tuber cumberlandensis now. Keep us moving along here. Truffles. Right. So when you think of truffle is definitely a gourmet delicacy crop. And we have our own native species. Now, tuber leonii was commonly known as the pecan truffle. And so when we received a donation of our trees, we thought that they were tuber leonii, the pecan truffle, which is another native truffle. But it turns out that the tuber genus isn't that well understood here in the United States. And what we have is actually a new species. It went under the name species 66 for a long time, but now it's called the Cumberland Mountain Truffle, tuber cumberlandensis. And it's a white truffle like you see here. This is a Perigord truffle, which we could also grow. So thanks to a very generous donation by Maker's Mark, UPike now will have one of the largest truffle research orchards on the East Coast, 3 acres. Not all of that will be under trees at first, but we'll grow into it. Um, so the Maker's Mark is calling this tuber cumberlandensis the Kentucky winter white truffle, and they value it around $70 an ounce. Basically, nothing is known about um, tuber cumberlandensis. We don't know what pH it prefers. We don't know really anything about it. We know that it grows on hazelnuts and oaks, which is what our orchard is comprised of. So it's mostly hazelnut, American hazelnut trees, and some oak. And it's going to take, we know it takes around 4 years if everything is perfect, up to 7 years before you start actually seeing, the tubers underground. So this is still in the discovery phase, and the industry needs a lot of help here, right? They need more people researching this, and thankfully, this is something that we can tackle at UPike. We're not truffle farmers, and so we don't have to worry about turning a profit in 4 or 5 years. And so we can set up these research orchards, take donation of these truffle trees, and then start testing some of these things, like what is the pH that they like? How much humus in the soil do they like? All those types of questions. And this truffle orchard is actually on a reclaimed strip mine, by the way. Let's see here. Okay, so I have a question. What are the truffles gaining from the tree, and does it benefit the tree? Yeah. So they're a mycorrhizal species, and so both parties are going to benefit from this. They're not harming the tree at all. They form mycorrhizae with the roots of the tree. We're going to talk about that in just a second, in a couple of slides, in a different sense. But basically, they form this association with the tree's roots, and they'll provide the tree with water and nutrients. And in exchange, the tree will give them glucose from photosynthesis and other phytocompounds. And so it's a very good symbiotic relationship here. And so you can actually harvest hazelnuts and the tubers, the truffles. Interestingly, the truffle harvest season is right now. And so there's a couple farms in Kentucky, and they're harvesting their truffles right now, which is really nice. Here's just an outline of the, doesn't give you too much context. This is on a main ridge line right in the middle of Pikeville, on a site that the university owns that we're calling Byron Mountains. This is the main ridge line. This is actually a power access road. And so this was all reclaimed mine. We've come in, we worked with Green Forest Works and a couple of others, some individuals from Georgia State, to there's no handbook on this, right? There's no manual for how do you put a truffle orchard in the mountains of eastern Kentucky. And so what we've done is we adopted some of the protocols for reclaiming strip mine land for nut trees, and that's what we've done. So this area now has all been cleared out, and we're actually in the process of fencing, putting up an 8-foot deer fence around this site. And our first block of trees will actually fill up a little square here in this bottom to middle section here. And I'll take questions on the truffles too. But let's talk about this NSF now. So the NSF, we're pivoting away from like the gourmet mushrooms that we typically grow. And what we're working on now is I have a second lab, and the second lab is a complete hydroponics lab. I have multiple hydroponic systems in place. It's all indoors, all under artificial light. It's all climate controlled. It's all automated. And it was quite the pain putting it all together, right? Because we built it all from scratch. Here we can see this is actually a germination rack, and what we see in the germination rack here is some peas here. So we grow microgreens and some other crops just in the meantime. So I have just a couple pictures of some of the crops that we kind of grow, and we're still getting the hang of this, right? We're mushroom farmers. We're not plant farmers, but we're learning. So we have some Salanova lettuce. We have some basil that we're growing. But right here is what we're really after. Okay, we're after the roots. We're after the roots. So this whole project, people always ask me, it's like, what's the point of the vegetables? It's like, well, we're actually not worried about the leafy greens or vegetables that we grow at all. I'm only really interested here in the roots. Before I go on to the next slide, I'll tell you the hydroponic systems that we have so you have an idea. We have one 5-rack ebb and flow system that's automated, and we use that primarily for germination. And when it's not in germination, we grow things like the lettuce and the basil and microgreens in it. So we have that ebb and flow system or flood and drain system. We have 30 Dutch buckets that are all daisy chained together that run through an automated dosing system. And underneath each of those buckets, so the buckets actually be right above where this picture was taken. We have lettuce rafts. You can see another one here. And the lettuce rafts are just that. They're just floating rafts that we're just trying to utilize all the space that we have, right? So we're growing lettuce in these. But what we're interested in is what's going on here in the roots. Okay. And so our portion of this ERISE 2 grant is we want to know what type of fungal biodiversity is in this rhizosphere area. So there's this really magical kind of unknown frontier of the rhizosphere, and it's where you have these root hairs and the rhizoplane here. You can see this is labeled out pretty well, but it's basically the contact face. It's the port or the docks of the roots of a plant, and it's where those roots contact with the soil. Okay, so you can think about a busy port in like New York City, right? All the goods being exchanged. And the same thing is happening in your garden. Okay, so you have all of the, nitrogen fixing bacteria, the nematodes, the fungi, the viruses even in it. So there's lots of biodiversity in this little tiny plane. And what we want to know is what happens if you remove the dirt? What does this biodiversity look like here if there's no dirt? So are the fungi that are they there from the seed? Are they coming with the seed? Are they in that embryo already? Or is it something that they accumulate just being in the dirt? Like are those fungi present in the dirt and then they form the associations with the root hairs as the plant develops? And we really just don't know, right? If I had to put some money down, I would imagine that the biodiversity is going to be quite a bit different between a soiled system and a soilless system in hydroponics. Um, Clearly, hydroponics still works because, I mean, there's lots of farms now that's their bread and butter is growing hydroponic soilless crops. So it still works, but would it work better if there was a difference between a soiled system and a soilless system? What if we could identify the fungi and other microbes present in a healthy garden? And what if we could inoculate hydroponic systems with those fungi and bacteria? What does that look like? And here's another example. Here's another example of kind of what's going on here. So you see all of these things, all of these microbes coming in to talk and exchange nutrients with the roots of these plants. And this is a big reason why most of the people in this chat are probably familiar with the difference between dirt and soil. If you plant your garden and you've got good soil, your garden does really well. You don't have to micromanage it too hard. But if you plant it in just kind of inert dirt, your plants are stunted, right? They don't work as well. And we're not going to get into all of these different things going on here, but I want to look over here at this AFN. This is arbuscular, mycorrhizal fungi, and so this is a type of mycorrhizae. So this is a fungus that actually gets into the plant cells and forms what we call arbuscules, these little pockets inside the plant cells. And these little pockets, if you can imagine each of these is like a loading dock inside of the plant cells, and things are being exchanged from the fungi. So the fungi is pulling it in from the outside into these arbuscule pockets, and then as things move through the plant cells, the plant and the fungi can interface and exchange things here. And so this is really what the NSF grant is focused on. All of those beautiful crops are just a bonus, but really we want to know who are the players in this game. And so what we do is we take root samples at various time frames. So, prior, just post germination, when the cotyledons are up, and the first sitting down their first roots. When they get their first set of true leaves, after a week, after 2 weeks, after 3 weeks, before when they go into flower. So we're really trying to get a full lifespan image of what these plants do. Now, in our Dutch bucket systems, um, we've grown somewhere in the ballpark of like 27 different things already in the hydroponic system, just getting a feel for it, right? Getting all the researchers trained on it. When we start processing the roots, our first species that we're going to look at is just going to be tomatoes, right? We have some tomatoes that we're going to look at, so we can get a baseline replicated system for just the tomato, and then we'll expand into other crops. I would really like to see, as this um, grant progresses, to look at more native crops. I would love to look at ramps and some of our native herbs and see if we could grow those in an accelerated manner. And then just another slide here. Right. And if you're interested, all of the sources for this are in the comments of these slides, and I can share the slides with you, so you can look at the sources. But again, there's lots of stuff going on here. There's lots of chemical signaling happening here, um, which is a whole nother area of the different pheromones that these things are releasing to communicate back and forth to each other. This you may have heard it referred to as the wood wide web before, um, where plants and things can communicate via these mycorrhizal associations. But yeah, that's really what we're kind of interested in. That's the end of the slides that I have. Oop, I got a comment. Is the exchange on demand? We don't know. I'll go ahead and stop share here, and I can answer some questions here, but we don't really know if it's on demand. It seems like in some cases with some species, it's on demand, and then there's these threshold barriers where once a certain chemical gradient is reached, then the nutrients will exchange. In some cases, it looks like the fungi can just take it if they want it. Um, Sometimes, it looks like the microbes can take what they want and vice versa. The plants can take what they want. So we really just don't know. We're still, like I said, in this discovery phase. We really don't know. We don't even know who the players are at the table yet, if that makes sense.
On the truffles, so I had a couple of questions. One,
Sure.
so Maker's Mark made a donation to UPike, presumably for research in
Yeah.
truffles, and they thought they were giving you one type of truffle, and somehow you discovered that it was not that truffle. So, did you identify that after you harvested and it didn't look right, it didn't taste right, or what cued you up that it wasn't what you thought you had?
Okay, so yeah, we haven't harvested any yet. Our trees are still about a year old, so we're still 3 years out from harvest. We didn't determine that they were a new species. We actually worked with the North American Truffle Growers Association, NATGA, and Maker's Mark. And how that got about was Margaret Townsend and Steve Townsend that own Townsend Truffery in Western Kentucky. I had them come to campus because I teach a course every fall called Mushrooms and Molds, the Hidden Kingdom of Fungi. Um, and it's really like an ethnomycology style course. And so I brought Steve and Margaret to campus and their lovely truffle dog, and we went on a truffle hunt in Pike County. We didn't find any. It was a bad season, they said, but after that communication, they reached back out to us and said that Maker's Mark was putting in a large orchard for their own use down at their I think it's Star Hill Farms, maybe is the name of their Maker's Mark's big farm in central Kentucky, and that they had some extra trees, and would we be interested in the extra trees? And so we jumped on the offer, and then things kind of went from there. We now have some other groups that want us to put different trees in with different species to look. But when we originally got the trees, um, everybody kind of knew that they were probably not tuberleonii. Um, we were going under the assumption that, hey, we're calling them tuberleonii right now, but it's very likely that they're a new species. I don't know. Oh, my kiddo turned off the light. Um, let's see who. I think maybe Tim Brennerman was the group that identified them. But yeah, it just come out in the last month or two that it's Tuber cumberlandensis, named after the Cumberland Mountains. It seems to be where they're at. And now we've seen that there's actually a couple places in Kentucky where truffle orchards who think that they're growing Perigord truffles. When they're harvesting, instead of getting black Perigord truffles, they're actually getting this Tuber cumberlandensis, which tells us that it may be better acclimated to our environment and actually outcompete some of these other somewhat more desirable truffles.
So how did it, if it's native to here, and
Mhm.
they brought these trees, and I don't know if it was pecans or filberts or what they brought you, but whatever it was, my understanding is that has to be pre-inoculated, and I don't know
Yes.
what stage that happens, but it had to have been inoculated somewhere else where they brought the trees from. So, um,
Yes. Yeah, it was inoculated. Sorry, go ahead.
Yes, but you're thinking that this thing is more widespread in Kentucky and that we already have it and that it's
Yeah.
growing on oaks and other things that we've got.
Absolutely. Yeah. It certainly is widespread in Kentucky, I would say. It's just there's not a lot of people going out and looking for them. These particular trees, they were inoculated as seedlings in somewhere in Lexington at a farm. I won't say the farm name just because I don't know if he wants it out, but they were inoculated and given to makers or sold to Maker's Mark, right? And so they were inoculated as seedlings, all hazelnuts, American hazelnuts, and then I think we had a few maybe pin oaks that came with the bunch, just to kind of space out the tree canopy a little bit. So we received them as bare twigs, and actually planted them in a little nursery plot for a year while we prepped our space. And so right now they're being held in a nursery plot while we get the fence up. And then, in the next month and a half or so, we're actually going to pull them from the nursery plot and put them in their permanent home up on Bear Mountain.
And
But
since
the individual did think he was inoculating tuber leonii.
okay.
Yeah, because what you do is you basically take the truffles. One of the ways is you make a truffle slurry. And so you take kind of the scraps of your other truffles, because when you break one of those truffles open, all of that gleba is what they call it, is just masses of spores. And they'll pulse it in a food blender and mix it with vermiculite, and make kind of the slurry and then dip the roots down in it is one method. I don't know if that's the method he done. There's several different ones, but we've inoculated our own using that method. And so, what I would wager, and I haven't confirmed this with the individual, is that when he made his truffle slurry, thinking they were tuber leonii, it was probably tuber cumberlandensis because they were present in his orchard.
You also mentioned the bioluminescent mushrooms that we have
Yeah.
locally, and um, I guess I was going to ask if there's ever an opportunity to do. I'm assuming that you go out on a night walk or something to see these bioluminescent mushrooms, and I wondered if there's ever something like that that you have like uh, an opportunity for the general public to come out on a nighttime walk to see these things. That would be
Sure.
really interesting.
I've never hosted a night walk before. Well, I take that back. Um, what I usually do is now I'm so I can see them so well during the day, and they don't look like much, right? They're little tan, kind of unassuming mushrooms. But once you kind of realize how, especially the Panella stypticus, once you learn what it looks like, you can spot it out while you're walking down a trail. And so for my students, what I usually do is as we're out field collecting, I'll grab some branches of those, and then we have a dark room at school, and I'll actually go in there because what I found, for Panella stypticus, it's maybe a 60 or 70% bioluminescent rate. Um, it basically means if, they have to have all of the bio pathway available to them, all the enzymes available to do the bioluminescent. When they run out, they're out. And so, like I said, about 60 to 70% of the sticks we bring back will actually glow. But if you look at something like a jack-o'-lantern, I find it to be much lower. My sample sizes on these are really, really low in terms of the jack-o'-lantern, so maybe it's a bit anecdotal. But the jack-o'-lantern, I've only, maybe 25% bioluminescent rate. But
I didn't know
um,
jack-o'-lantern was bioluminescent.
oh
And
yes, yeah.
the genus and species name for the other one that you were mentioning, is that foxfire or is it something else? Because I don't recognize the name.
The Omphalotus.
You rattled one off that I couldn't repeat it if you said it to me, but uh, um, so I guess let me just say it's do they pulse? Where I was going with this, I've seen Foxfire before, and it pulses. So it's not
Yeah,
uniform brightness. It'll fade and it'll almost disappear, and then you'll
yeah.
see it build back up again. Do all of these do that same thing? They kind of pulse.
Yeah, a little bit. They're really kind of eerie when you first see them coming out of the darkness. Because usually, we'll go in the dark room and we're standing there. Really kind of silly. You're standing in this dark room with a stick in front of your face and you're wanting it to glow so badly, and then it kind of starts emerging from the darkness, and you think, "Am I seeing stuff, or is it actually glowing?" But it does have a little bit of a pulse. And what happens here is there's a luciferin pathway. The what actually makes it glow is there's a compound called luciferin that an enzyme luciferase comes and acts on it, and it's an ATP pathway, and it produces this glow. And so the naming behind it's actually really interesting. They named it luciferin after Lucifer, which ironically means bringer of light, right? And Aristotle called it cold fire. Because the Greeks, when they seen this, is like, here you have a light, but it's not producing heat. It's cold fire. And then the French took that term and said it's faux fire. And then us Americans took the term faux fire and turned faux into fox. And so you get foxfire. And foxfire is probably multiple species that are all kind of masquerading as one thing. Um, I've seen people say that the jack-o'-lantern is foxfire, but I've also seen reports that what's it called? Honey mushrooms, armorillia,
Oh, they're
armorillia.
really tiny. The
No,
ones
these
I've
these
seen
can
are
be a little bit
really
big.
small.
Oh
Uh,
yeah, these are Mycena, not Mycena, sorry, Pinella stypticus. They are really small. Now, the armorillia, the mushrooms don't glow, but the mycelium does. And so you'll get, leaves that'll have these veins that are glowing. And all of them kind of have a different intensity. They all glow more or less the same wavelength of green, um, but will have different intensities. And they also seem to have a bit of a circadian rhythm to them. So at nighttime, they'll glow more, I've noticed. When it starts getting towards the evening, the bioluminescence tends to pick up. And then during the day, it seems to taper off a bit. And so,
and is
the
that an attractant or is it just a coincidental side effect of something else that they're doing?
I
Does it
think
occur?
that it's probably just evolutionary baggage. Um, I don't think that's an attractant. There've been lots of studies that come out that say, is this an insect attractant? And while it's possible that it does attract insects, um, I wonder how beneficial it is if your mycelium underground is glowing, right? Maybe you're attracting nematodes or something with photoreceptors that way. But if you look at the case of a jack-o'-lantern mushroom, I mean, that mushroom is going to produce hundreds of millions of aerial spores. You have hundreds of millions of potentially germinating particles that get dusted through the air. So how beneficial could some insects getting to it a little bit sooner be, right? Um, and so, I don't know. There's studies that show that or suggest that it could be beneficial. Um, and some studies seem to suggest that maybe you know, it's it's a good way for them to metabolize byproducts that they're making, and generate some energy, but not a lot of heat. So I think that the verdict is still out on why they do it. It's just a really cool feature that they have.
And these are medicinal. So what are the medicinals that you're isolating from these?
Uh,
Or how would they be used?
so we're still looking at the medicinal compounds in Pinella stypticus. Pinella stypticus seems to have a couple of compounds that maybe are tumor suppressant. There've been some in vitro studies that are still early on that seem to suggest that they're tumor suppressant, but we just don't know enough about it yet. And so I'm working with a biochemist, Dr. Shihong, at UPike, and she's a medicinal biochemist, and so she's looking at those extracts. Um, I'm not too sure what their properties are outside of there may be some tumor suppressant benefits there. Now, the Omphallotus also appears to have some medicinal benefits, but the Omphallotus also has some toxic alkaloids, right? And so several people every year get, or maybe dozens of people every year get poisoned accidentally by jack-o'-lantern mushrooms. And so there's certainly a lot of biochemically active compounds being secreted by these fungi. And so which ones are medicinal and in what dosages too are they medicinal is still we're still learning.
All right, I'll shut up now and let somebody else ask. Anyone? Surely I wasn't the only one that had a question.
See, the teacher in me, I can't see everybody's eyes, and typically when the crowd falls silent, I look for eyeballs, and then they get volunteered, but I can't see eyeballs. So
Well, I thought Chris or Phil or somebody would chime in um, and ask something. So very interesting talk. We do reishi at the extension office in Whitesburg in a grow chamber, which I think Amazon has like a $300 grow chamber that I'm pretty sure originated from some other use that I won't go into, but
Absolutely. Yeah.
uh, it controls the humidity and has the grow lights and vents the air. And I didn't understand. You taught me something tonight. I didn't know that they used oxygen. I assumed that it was a CO thing like the plant. So I wasn't sure why they needed to vent. And we have shiitakes in there. And you know, when shiitakes vent, it's garlic. It's
Mhm.
his office. It's my assistant's office. The whole office smells like garlic. So uh, pretty pretty fun thing to do.
Yeah, absolutely.
Hey, Byron, good information. Jeremy Williams, ag agent over here in Harlan County. Good information. I really like the work you're doing on I guess the simulation mushrooms in the coal mine. Uh,
Yeah.
I had some great success on shiitakes several years ago, putting them in on logs in a mine, and
Okay.
it was amazing. And so I think that's a great opportunity for us here in Eastern Kentucky, Southwest Virginia. So uh,
Absolutely.
I'm interested in that data that you're compiling there because the success that I had, there was a lot of potential, and there was actually a paper written in the '60s. Like you said, it's not a novel idea. Paper written in the '60s of a coal mine in West Virginia that had received some federal dollars, actually, if I'm not mistaken, doing that. So I'm looking forward to seeing what you come up with. That's awesome stuff.
Yeah, thank you. Thank you, Jeremy. Yeah, all of that data will come out in the report. I'm happy to talk more about it. One of the things that we want to do with the USDA grant is to actually come out and train folks and have these conversations with folks. And so if it's something your agency is interested in doing, we have inoculum, we have all the stuff here. And what I didn't mention is most of our strains, we actually have an Appalachian genetic library. So we bring in all the Appalachian genetics that we can. And so it seems like I don't want to reinvent the wheel, creating new strains of mushrooms, new cultivars, when they've already been here for eons, and so they've already been acclimated to this environment. So let's use what we have, and then go from there with our selective breeding. But yeah, I've seen a couple of people have mentioned the paper in the '60s. I've not seen that, but I'm hopeful that if you have it or if you know where I could get it, I would love to get a hold of it and read it. But we're getting people now. We have a partner out in Colorado that has a precious metal mine that wants to grow mushrooms in it. We were approached by a college in Norway that wants to grow mushrooms in their coal mine in Svalbard, which is the northernmost. It's it's north of the Arctic Circle. Um, and so lots of places have similar issues that we do. It's like, we need more food, better food, locally grown, that's still fresh. And I think this is a perfect food crop that we can grow in the mountains.
Yeah, Jeremy mentioned the one they had in Harlan, and I was going to say that the folks that had the one in the bathhouse,
Yeah.
um, that was across the way from Portal 31. One thing I noticed is I believe it was totally dark. I don't know if they provided light in there or not. Maybe they did, but when I went in, there were secondary things growing in there that I reacted to. So black mold or something, and
Yeah.
I could feel it on my throat when I walked in. So how do you control if you do this in a deep mine or somewhere else? How do you control those secondary fungi that are maybe counterproductive?
Yeah. Yeah. So, I mean, you are creating an environment that fungi will thrive in. And so you control that through rigorous cleaning all the time. I mean, we're always, always, always cleaning. Um, every day that our chambers get cleaned down. Inside of mine, the easiest way would be to break it up into sections and clean those sections. Now, if you're bringing water in, if you have to humidify the air, it's not that bad. We use a UV sterilizer inside of the water containers, and so that helps us. And we have a reverse osmosis system, so our water is already pretty pure when it goes in. But yeah, you'll get mildews that want to form if you're not regularly cleaning. You want to make sure that you're harvesting the mushrooms before spore drop. And so when you think about a mushroom cap, as it starts to unfurl, if it gets all the way unfurled, it's going to drop spores and a lot of them. And so you really want to harvest them when they're still got some concave to them. And that just takes practice. But really, it comes down to having a pretty robust cleaning schedule and making sure that you're staying on top of that aspect.
The ones that I raised in the mine, I don't remember having a lot of issues with non shiitake,
Yeah,
fungus on it. It had been doing due to the ventilation. It may have been more controlled. It was one of those things. The only issue I had was at times there was lighting issues. Certain times of the year there was lighting issues of basically no or very little light. Whereas other times of the year the lighting was perfect. So
yeah,
getting light, there's sometimes a year I was getting light 8 hours a day. So other times of the year I may not get very little light. But no, I don't remember a lot of issues. And it may have just been where it was so controlled.
yeah,
And the ventilation was good.
So
yeah.
how are they
Certainly,
benefiting
mine.
from that light? Um, Light seems to be a factor in it, but they're not producing chlorophyll. So what exactly is the light doing for the mushroom? How is it gathering that?
Well, they have other pigments, so they have melanin pigments in their cells, and so they'll their color will intensify quite a bit. They don't really need much light. They're phototropic in the sense that if you give them light, it helps them know which direction's up, but they're also slightly gravitropic, and so they can already sense which way is up anyway. But if you give them light, it'll actually help stimulate the pins to form. And so really, if you wanted to maximize the amount of light you give them, if you give them some light to start the pin set, you could probably dial the light back. Right now we give them, that picture of my chamber that was with the lights on, but typically I don't have much lights on. I have lights that you might think of in a mine. It's just some kind of lights here and there. More or less, you can read a book in there, but that's about it. Um, but there does seem to be some research to suggest that red light and blue light at different phases of their life may increase pin set or increase fruit density. If you want really brightly colored mushrooms, give them a lot of light. So those gold oysters, pink oysters, blue oysters, if you want them to be really colorful and pop, give them a nice full spectrum light. Um, I don't think that you need to prescribe the red versus blue light. Full spectrum seems to work. It's not 100% efficient as compared to if you're splitting wavelengths, but it certainly works. But something like enoki, our native strain of enoki, Flammulina velutipes, the velvet shank, the velvet foot, in the wild, enoki is really kind of a squat mushroom that forms these little dense clusters. But if you go to, if you were to go to Asia or go to an Asian restaurant and order ramen or something, you get those long stringy mushrooms in your noodles. That's the same species, right? It's just that culturally they want the mushroom to be longer and they want it to be white. So what they do is if you want it to be white, restrict the light, don't give it any light. And those pigments won't activate and it'll stay a white mushroom. To make it long, they put these collars around, they grow mushrooms in jars. And so they put this collar around the jar. And what that does is it keeps the CO2 really high inside that jar. And the mushroom continually stretches to try to get out of that CO2 environment, right? But here in the wild, they grow on the sides of logs, and so you get these really short, big caps, kind of short stems, and they're orange and black.
So
And
they
so,
get leggy just like a plant gets leggy
yeah,
in low light conditions.
yeah, exactly, right. If you leave that plant in germination just a little too long in the dark, right, you pull the tray off, and now suddenly it's leggy, trying to run on you.
Hmm. Very good. Anyone else? I think we had a comment. Let's see what we saw. All right, very good, Debbie. Byron, I'm assuming you can see that.
Yeah, thanks, Debbie. We're certainly um, we're very excited and appreciative that we're being able to do this work here. And that, a lot of times students think they have to leave the area to do this type of research, and they don't, right? Like I said, I have 17 undergrads. They're all paid, and they're paid very well. They're paid, in all of the grants that I write. the bulk of the funding goes to student researchers because I want to keep our talent here. I don't want our talent to have to leave the state or to go to the other end of the state in order to find meaningful research projects that are going to help them in their future careers, but also just help put food on their table. And so, I'm really thankful that we've been able to fund these students and get them involved on this research. I certainly couldn't do any of it without them.
And you also mentioned lichens briefly in your talk, and uh, Phil and I and Jeremy have had an interest in um, a lichen workshop just because we've never had a presentation on that, and
Yeah.
I can't speak for those two guys, but I could say that I'm I recognize what they are, and I know that there are multiple species of lichen.
Oh yeah.
And we've got some of the mint green ones, and then we've got the big black and drab, olive drab, flaky ones that like to be on Pine Mountain on the rocks.
Oh yeah.
And so, uh, if there's ever an opportunity to get a talk in on lichens from you or someone we would appreciate that.
Yeah, absolutely. I'd love to give a talk on lichens. We actually, uh, every year, the Breaks Interstate Park hosts a foray. They're doing it in the spring this year. I can send you the dates. I don't know them off the top of my head, but they're doing a spring foray from the Central Appalachian Mycological Society. And I'm a co-founder of that society. And they have lots of great walks at the Breaks Interstate Park. And in the past, I ran several walks. I do a lichen walk there. They do a bioluminescent walk and a fluorescent walk.
Oh, wow.
Um, yeah, so they do a lot of. I mean, last year we done it in the fall, and um, we probably had 100, 150 people show up, and we collected somewhere in the ballpark of 4,000, 4,500 species of fungi. It's the
Wow.
only time that you're allowed to collect at the breaks. And I have a collection permit that I pass on to the group that comes. And so we kind of collect everything and catalog it. I have two students that have went to be rangers at the park, um, at least seasonal rangers. And I have one right now. It's a bumblebee guy. He's done a lot of work on bumblebees. But they do walks. I've trained them on how to do the lichen walk. So the breaks very often throughout the season does mushroom walks on the weekend. So they'll do slime mold walks, mushroom walks, lichen walks. And so if you guys are interested in that type of programming, the breaks almost always does that um, on weekends during season.
Yeah, if you could send that out, that would be great. We'd
Yeah.
like to advertise that. I've never noticed that they do that.
Yeah. Yeah. I
So,
mean, it may be one of the only parks that kind of does those type. I think maybe some parks in Central Kentucky now are starting to do that, but
okay. Probably the only question we get from Extension, other than uh, growing shiitakes or something like that, is the Gnostic. When we have wet years and
yeah.
it's in everybody's driveway and they're wanting to know, what do I spray to kill this stuff out, or what do I do to get rid of
Yeah.
this? And we do occasionally get questions about it.
Yeah. Yeah. Gnostic's a cool one. Um, and and sometimes the algae that helps form Gnostic will actually help form lichens. Lichens are really weird. They're their own thing, and they're really cool. Um, we have, gosh, if you have what I call a good tree, outside and it's covered in lichens. I had a mentor one time that said he was a botanist, a very well regarded botanist in the state, and he said he would joke and say, "I don't know what tree bark actually looks like," because when you go look at tree bark, all those different colored mosaics, those are all lichens. And so one good branch could have 20, 25 lichens on it. So they're here in abundance. Um, all of the kind of cryptic life we have in bushels. There in Pike County alone, there's something just north of 400 species of moss. So it's just we have biodiversity coming out of our ears.
Right? That's great.
Yeah.
Anyone else? All right. Well, Byron, thank you for taking the time to speak to us tonight, and
Yeah.
wonderful talk, and lots of information. And we encourage you to continue what you're doing at UPike, And hopefully, as there are opportunities, we'd like to maybe pop down and visit sometime and see what you're doing.
Yeah,
Be a great tour
absolutely.
for the Ag Agents Association to come down and see that. Uh,
Yeah.
and if you
Yeah.
get that information about the walks, that's great too.
Yeah. I'll add in real quick before we finish here that Dr. Crocker and I are actually working on her and I are working on some trainings, mushroom identification trainings to roll out to everybody, and maybe in the future cultivation workshop trainings. So I'm working with Dr. Crocker on getting some formalized material out for everybody. But you're all certainly welcome to pop in at UPike if you have questions. Shoot me an email, even if it's non-mushroom related. If you have biology questions, shoot me an email. I'm happy to respond, and we can chat.
Very good. Well, thank you very much.
Yeah, thank all of you for coming out.